Carbohydrates are important in many more places than you would think. Everybody knows that carbohydrates are behind sugars and honey and starch and grains. But did you know that sugars also build cellulose and chitin and bacterial cell walls? And joined up with proteins make up cartilage and mucus? The word carbohydrate is a funny old-fashioned one, and it comes from the fact that the empirical formula for simple sugars is CH2O n times, as if it was made directly from carbon and water. Simple sugars are built from multiple of these HCOH units like that, and we don't normally call them carbohydrates until they've got at least 3 of these units. The most common ones in nature have between 3 and 6 carbon atoms. One of the ways of classifying simple sugars is by the number of carbon atoms, so those with 3 carbon atoms are called trios, those with 4 tetros, those with 5 pentos, and those with 6 hexos. You will see later that simple sugars can join up to make short or sometimes very long chains, and to make it clear that these are the smallest sort of single individual units, we call them monosaccharides. There's one thing we've overlooked so far. All monosaccharides have one carbon atom that is more highly oxidized. So instead of carrying a hydroxyl group, the carbon atom is oxidized to the level of aldehyde or ketone. These are actually the same oxidation levels, but we get an aldehyde if the first carbon atom is oxidized, and a ketone if a carbon atom that's not at the end of the chain is oxidized. And almost always that is number 2. Apart from the number of carbon atoms, monosaccharides can also be classified according to whether they're aldehydes or ketones. We call them correspondingly aldoses and ketoses. Confused? Let's try and get some order here. We said that the smallest carbohydrates are the trioses with 3 carbon atoms. If that triose is an aldose, it looks like this. Note that the aldehyde group is on carbon atom number 1 in this chain. And that's actually not a coincidence. By convention, the most highly oxidized carbon atom gets the smallest number, so it would not be 3, but is 1. Now, there's a 3-carbon ketose as well. In this case, it's carbon atom number 2 that is oxidized. Great, so now we've understood that. The bigger monosaccharides are just like extensions from here. So there are aldoses with 4, 5, and 6 carbon atoms, as well as ketoses with 4, 5, and 6 carbon atoms. This looks a lot more complicated than it is. Why not just pause the video right here and take some time to have a closer look? Alright, that wasn't so bad, was it? But unfortunately, there's one more thing that makes everything a bit more complicated. Let's have a look at that first aldose again. This is a triose called glyceraldehyde. You might remember that the first carbon atom with its double bond to the oxygen has a trigonal planar geometry, while C2 and C3 are tetrahedral. So in 3D it might look something like this. Let's check. The aldehyde group is on top, and then 1 hydroxyl group each on C2 and C3. Great. But what about this one? All the functional groups are in the same place. So the aldehyde group and the 2-hydroxyl group, but the 2 molecules are not identical. If you turn the one on the right around, you can see that it is actually a mirror image of the molecule on the left. We call pairs of molecules that are mirror images of each other enantiomers, and molecules that have this property of coming in 2 or more forms that can't be superimposed by rotating them, chiral molecules. That word comes from the Greek word for hand, which you probably know from the word chiropractor. So in English we'd say the molecules have a handedness like a pair of gloves. This always happens when there are 4 different atoms or groups bound to 1 carbon atom. And such carbon atoms are called asymmetric. All chiral molecules have at least 1 or sometimes several asymmetric carbon atoms. If they have 2 or more, it gets pretty complicated, and we'll see that later, but that's for another video. For glyceraldehyde, both the left and the right form exist, but in nature we mostly find the form on the left. So how do we even know which one we're talking about? What we need to do is to agree on a way of showing or drawing the two structures and assigning them unambiguous names. First thing we do is to rotate the molecule so that the carbon chain is vertical, and the most highly oxidized carbon atom, which is the aldehyde group here, on top. We also need to make sure that the asymmetric carbon atom we're looking at is in the foreground, and the neighboring carbon atoms a little further back. That way we have the two other groups, here the hydrogen and the hydroxyl group, pointing left and right, and a little bit towards us. Here are the two forms of glyceraldehyde again, oriented the way I've just described them. Let's draw these structures on top. As you see, by convention we use these dashed bonds to indicate that something is pointing behind the plane of the screen or paper, and the solid black wedges for atoms or groups that are pointing more towards us or that are above the plane of the screen. If we can agree on those conventions, then we can draw the structures much more simply, like so. The top structure is called D-glyceraldehyde, the bottom one L-glyceraldehyde. The letters come from the Latin words for left and right, but it's probably easiest to remember that L is for left. This style of projecting the 3D structure onto a 2D paper or diagram is called Fischer projection. Wow, take a deep breath. Why not pause the video here and take a closer look at how 3D and 2D illustrations relate? Alright, so we've seen that glyceraldehyde comes in two forms, D and L. One asymmetric carbon atom, marked by the red asterisk, means that there are two forms, or one pair of enantiomers. But what about the bigger monosaccharides? Tetroses, the C4 sugars, have two asymmetric carbon atoms. And because there are two configurations for each of them, there are 4 different tetroses. That's 2 pairs of enantiomers. These go by the same name, so there's D and L erythros and D and L threos. If you've got eagle eyes, you might have noticed that D threos has 2 hydroxyl groups pointing in different directions. So why is it called D and not L? Well, it's because, by convention, it's the position of the hydroxyl group on this carbon atom that counts. This is the one that is the farthest away from the aldehyde group. In other words, the asymmetric carbon atom with the highest number. That sounds a bit arbitrary, but it's actually not, because in nature we find almost exclusively the D forms of sugars, and there's no coincidence because their biosynthetic pathways are all linked. So, if there is 1 D triose and 2 D tetroses, we would expect, 4 D pentoses and no fewer than 8 D hexoses. And that's just the aldoses. Let's remember that there were ketoses as well. Many of these sugars are actually very rare in nature, but down here we find, for example, glucose and galactose. So let's wrap up. We've seen that simple sugars, the building blocks of all carbohydrates, really are built from multiples of a CH2O unit. We've seen that they are called trioses, tetroses, pentoses, and hexoses if they have 3, 4, 5, and 6 carbon atoms respectively. And aldoses and ketoses depending on whether the first or second carbon atom is oxidized. Almost all sugars are chiral, which means they have asymmetric carbon atoms that come in 2 different configurations. A chiral molecule pair, where one is a mirror image of the other, is called enantiomers. And finally, there is a convention, the Fischer projection, for writing down the structure of these chiral sugars, and derived from that a convention on which ones to call D and which ones L. The D forms of all sugars is by far the more common one in nature.